Most Pierre Changeux világhírű francia neurobiokémikus professzor úr előadása következik, azonban előtte felkérem Gulyás Balázs elnök urat, hogy röviden mutassa be a professzor urat.
Pierre Changeux, azt hiszem, az élő neurobiológusok doyenje, és talán itt be is fejezhetném a bemutatásomat. Ő az, akit neurobiológusoknak nem kell bemutatnom. 1936-ban született, a École Normale Supérieure-ben végezte tanulmányait, majd a Pasteur intézetben a doktorátusát 2 Nobel-díjasnál, François Jacob és Jacques Monodnál végezte. És már 25 évesen olyan világra szóló felfedezést tett, amely a mai napig is az ő nevéhez fűződik. Felfedezte az allosztéria jelenségét, az allosztérikus protein mozgásokat, jelenségeket, az allosztérikus enzimeknek az első komoly modelljét és leírását adta 25 évesen. Majd következtek ugyancsak világra szóló felfedezései, az acetilkolin receptor felfedezése, annak leírása molekuláris biológiai szinteken, és ettől kezdve számos világra szóló felfedezés az agyról, az agy működéséről, az agy kémiai, molekuláris biológiai alapjairól. Nem csoda, hogy messze többször, mint 100 000-szer idézték, utána lehet nézni, és 170-es hírsindexe van, ami hát azt hiszem, hogy önmagában is mond valamit a teljesítményéről. De aminél sokkal fontosabb, tevékenysége gyakorlatilag abból a szempontból is világra szóló, hogy összekötötte a molekuláris biológiát a neurobiológiával, a neurobiológiát a kognitív tudományokkal, sőt, ezen túlnyúlva még az evolúcióbiológiával is. Az a kutatás, amit ő folytatott, és folytat a mai napig is 87 évesen, mint aktív kutató, a neurobiológiának egy olyan keresztmetszetét adja, amelyet egy kézben nagyon kevés kutató tud kézben tartani, és ő tudja ezt. Összekötve a legkülönbözőbb területeket, egységes rendszerbe hozva, és így nem csak elemi neurobiológiai jelenségeket tud magyarázni, és magyaráz, hanem a legmagasabb kognitív jelenségeket, a tudatot is. Könyvein generációk nőttek fel, és az a fajta bölcsesség, amely őt ismerve belőle árad, az rengeteg kutatót termékenyített meg, és gondolkodtatott el, hogy mi a kutatásnak, ezen belül is az agykutatásnak, az értelme, a lényege, és hogyan kell tovább lépni kutatásainkban. Még csak egy dolgot, hogy nem csak kutat, zenél is. Lakásában egy olyan orgona van, egy pipe organ, tehát egy igazi orgona, amely egy egész szobát betölt, és ott játszik minden nap. Emellett természetesen filozófiával is bőségesen foglalkozik, erről is írt könyveket, és végül, de nem utolsó sorban az etika az az egyik rendkívül kedvelt és szeretett területe, évtizedekig volt a francia bioetikai tanács elnöke, és rengeteg megdöbbentően érdekes, kihívó és elgondolkodtató problémát, kihívást tárt fel előttem is az elmúlt évtizedekben a bioetika és a kutatás határterületeiről. Pierre Changeux, ahogy említettem, az egyik ikonja a neurobiológusoknak, és most itt van ismét Magyarországon. Nagyon szeret minket, az elmúlt 40 évben számos alkalommal járt itt, és öröm a számunkra, hogy ismét itt van velünk, úgyhogy szeretném visszaadni a szót a Master of Ceremoninknak, és kérem, hogy szólítsa Pierre Changeux professzor urat a színpadra.
Így van, köszönöm szépen. Most felkérem Pierre Changeux professzor urat, hogy tartsa meg előadását. Professor Changeux, I invite you to the stage to deliver your presentation.
Thank you very much, President Balázs Gulyás, dear friends, dear colleagues. This is for me a real honor to speak at this Science Day, which, to my understanding, is unique in the world, in Hungary. And also I have a great pleasure to talk about a topic which is the chemistry of the brain as a chemical machine. And this is, in fact, a very interesting topic because Balázs Gulyás was initially a brain imager, and I met him in Sweden when he was working on the brain imaging of the visual system with Pierre Roland. And at the same time, I was working on the molecular biology of the nicotinic receptor. So he was on the brain scale, I was on the molecular scale. And I may say that I am happy to celebrate today. In fact, the common feature that we have now together to work on the brain as a chemical machine. So this is a theme which for several of you may surprise, because usually one have problems to talk of the brain as a machine, and even as a chemical machine more. So this is the work of decades of research in the past with Pierre Changeux, Marc Delarue, Marco Cecchini, Marina Picciotto, Sylvie Granon, Yves Massos, and last but not least, Stanislas Dehaene. So I will subdivide my presentation into four main sections: the brain and its chemistry, receptors as allosteric regulatory protein, and allosteric drug design. Then I will deal with two issues which are complementary at a higher level of organization. First, at the level of synapse selection, and last, about consciousness and mental disease, if I have time. So the first thing I want to insist on, since in this room maybe there are non-neuroscientists, is that since the 16th century, we know about gross brain anatomy. It's an organ in our skull of about 1.4 kilogram with right-left asymmetry, many convolutions and fissures. And this complexity of organization is also shown by the illustration that there is gray with white matter. And most of all, just an example, because of the specialization of some cortical areas, up to 180 per hemisphere, including 97, which are new and unique for the human species compared to the monkey. So this process of complexification of the brain happened during hominidization, about 1 million years from now, started with Australopithecus, followed by Homo habilis, and then Homo neanderthalensis and Homo sapiens. And during this hominidization, there is an increase in size and volume, but more than that, compaction, number of cortical area, and complexity of the connectome. So this connectome is in fact due to the fact that the brain is made up of cells, neurons, as you can see here, and on the left, and these neurons show expansions, dendrites and axons, which are connected one with the other. So the whole brain is in fact an immense network. And one of the most important features that Ramon y Cajal found was that this network is discontinuous. And this is due to the fact that, as you can see here, when a nerve ending reaches another cell or another synapse, then there is a gap in between the nerve ending and between the postsynaptic membrane. There is a synaptic gap that one has to cross if you want that the information, essentially electrical, can go from one cell to another. And therefore, this discontinuous network implies that there is something involved in the relationship between networks, between cells in the network. And just following this very general statement, I may say that in the human brain there are up to 80 billion neurons and about 1 million billions of synaptic contact in the cerebral cortex. This is clearly the most complex organ of the body, and I may even say the most complex organ in the living world. So the synapse, which is this contact between nerve cells, as I say, involve a mechanism for transmitting signals from the presynaptic level to the postsynaptic level. And in most of the synapses, not all, but most of them, then there is a chemical compound called a neurotransmitter, which is involved in this communication. And there is therefore what may be called a chemical communication, thus the word chemical network, because all the neurons have to go through some kind of communication of this style, and there are about 20 neurotransmitters already known, as you may have heard about acetylcholine, about norepinephrine, dopamine, GABA, and so on, and many peptides. So this means that we are dealing with a highly diversified chemical system of intercellular communication, which make the brain network so complex, so huge, in fact, a chemical machine. So the question soon became important to try to find at the molecular level how this chemical communication works. And very early on, this was helped by pharmacology. Here again, applied science has an impact on basic science. And people were looking at the mode of action of drugs, in particular John Newport Langley for nicotine and curare, and struck by the effect of these substances, these compounds on, for instance, the fall. Langley postulated that there is I cite the word, the muscle substance which combines with nicotine and curare is not identical with the substance which contracts. I have called it the receptive substance. So this is an interesting point, and he added it is localized under the nerve terminals, but as we know, they may be outside the synapse from the work done here by Professor Visi. So this issue of receptor was raised in 1900, and concerning drug design, since we have to understand how these drugs act on these receptors, it was proposed from early on by Daniel Beauvais and James Black that in fact drugs should be compounds which bind to the same site as the physiological ligand. Let's say at the neurotransmitter binding site, then there would be acetylcholine and curare or galamine which would compete with each other. And Sir James Black clearly shown that with the identification of propranolol, which is the most currently used drug, as the first competitive blocker of the beta-adrenergic receptor binding site. So this then many neurotransmitter receptors have been identified, and there was a proposal of the concept of allosteric modulation. Now, Professor Balázs Gulyás mentioned this word, allosteric, and perhaps many of you don't know what it is. And I'm just going to spend a few minutes to tell you about allosteric interaction. It came from the original observation of Umberger and others that in bacteria there are a network of regulatory signaling which are taking place. And here on the left, I illustrate one of them. There is clearly an effect of isoleucine on the first enzyme of the pathway, threonine desaminase. There is a feedback mechanism as postulated by Norbert Wiener initially. So, starting my thesis work in the laboratory of Jacques Monod at Institut Pasteur, the first thing I was puzzled by this discovery was what is the mechanism involved in the effect of a feedback inhibitor on first enzyme. Is it going to bind to the catalytic site or not? So I was initially involved in the demonstration that you can find conditions where you keep the enzyme active and lost the regulation. And this same finding was also done by Gerhardt and Pardy on a different enzyme. And Jacques Monod then asked me to write a paper by myself about this discovery and to have it published in the proceeding of the Cold Spring Harbor meeting in 1961, where I first mentioned this work. And in this paper, you can see the two different kind of models that I presented. Of course, model 1 is a classical competition by steric kinase. I just mentioned it with James Black and Daniel Beauvais. And now, the alternative mechanism I suggested, model 2, was that there were distinct binding sites for the substrate and for the regulatory signal. And this was named, instead of model 2, which is a very non-sexy kind of denomination, it was proposed the word allosteric in the discussion of the meeting by Jacob and Monod, this is indeed the word which has been successful in qualifying this second model. So how could they communicate with each other at the level of a protein? Of course, the issue was there should be a conformation change. And at the end of my thesis work, I propose to make a synthesis of all the work I had done in my thesis, and in particular to study some cooperative effects which were found with this enzyme, as they are found with hemoglobin and oxygen binding. You all remember the S-shaped curve of oxygen binding to hemoglobin. So here we have the same phenomenon, and the postulate was that the protein should be an oligomer made up of several subunits carrying the active site, and that the link between the two would be some kind of transition between, I would say, a relaxed state and a tense state, where the interaction between the subunits would change during the transition. The other postulate which was made is that these states would pre-exist to the binding of ligand. And this is also an issue which is important, because classically pharmacologists think that if a ligand binds to a protein and have a conformation change, then it will be induced by the ligand. Here it is not going to be induced, but the conformation would be differentially stabilized by the ligand. And if I insist on that, is that simply because I am going to briefly discuss that issue in the case of receptor. So this was the end of my thesis work. Of course, there are still many studies done on allosteric proteins. And already in the conclusion of my thesis work, I suggested that this kind of mechanism could also play a role in the nervous system, and in particular in the brain. But there was no receptor protein identified at that time. So I went to David Nartmanson laboratory in Columbia, learned to work on the electrophysiology of the electric organ, and became familiar with the richness of this electric organ from fish in synaptic components, and in particular, eventually, of receptor. And to label the receptor, I use a toxin, a snake venom toxin, which was identified by Shen-Yuan Li, with whom we cooperate to identify the receptor, purify the protein, and have it observed for the first time. This is the first time, if I may say, in the history of receptors that a receptor is seen, purified on the left, and in the postsynaptic membrane on the right. And already you can see that several of these rosettes are made up of 5 subunits. This was further investigated by Carlin, by Raftery and others. And indeed, we are dealing with a pentameric receptor, which is a rare kind of oligomer. And then we went to identify the sequence. We made the first 20 amino acid sequence, which was followed by Raftery. We made a complete sequence of amino acids from amino acid sequence technique. Then the cloning of the mRNA was done, and the full amino acid sequence of the RNA was done, and the model which we proposed from the inferred amino acid sequence clearly shows that we propose that there is a neurotransmitter binding site element in the synapse cleft and a transmembrane in red segment where the ionic channel could be present. And indeed, what this receptor mediates is the opening of an ion channel upon the binding of acetylcholine, which I already shown in the previous slide. So already from this modeling and further demonstrated by image of Nigel Unwin by low resolution electron microscopy, one can clearly see on this picture that the acetylcholine binding site are distant by 40 angstrom, and that the distance between acetylcholine binding site and ion channel are 60 angstrom. So we are clearly dealing with an allosteric protein, since we have topographically distinct sites for the neurotransmitter and the ion channel. So this is, in fact, if I may say, a new era in neuroscience, the molecular biology of receptor, which has now developed enormously throughout the world and throughout through different neurotransmitter receptors. There are hundreds of neurotransmitter receptors which has been identified. Now, we wanted to further study the acetylcholine receptor at the atomic level. And to do that, we needed to have the X-ray structure of the nicotinic receptor, or an homologue of the nicotinic receptor. And I was, in fact, extremely interested by a paper which was proposed by Sasnim et al., which is just a paper on the comparison of various sequences in bacteria with those in eukaryotes, pure sequence work. And they show that there are, as you can see here on the left, several sequences which resemble a nicotinic receptor in these bacteria. And this was further documented, and we may demonstrate, for instance, from this receptor from Glioblastoma violaceus, which is a bacteria from Switzerland, from the mountains of Switzerland, that one of these sequences lead to the expression of a proton-gated ion channel. You see the patch clamp in Neher-Sackmann technique on the right. And we were able to purify it and hopefully to crystallize it. And therefore we have here a quite interesting conclusion about the origin of our brain receptor, at least for the pentameric receptor. The origin is bacteria, about 3 billion years old. Now, following this early discovery with Glick, and nearly at the same time, but not at the same resolution, by Hilf and Dessler from another bacteria, since these early studies with bacterial receptor, there have been several excitatory and inhibitory receptor which has been identified, GluCl, GABA, glycine, 5-HT3, and by Hips and collaborator, the nicotinic receptor, alpha-4 beta-2. So I think we have to assume that in our brain, we have receptors which are highly similar to the receptor in bacteria and work quite similarly. Maybe this is something you did not expect from my talk, but it is true. Now having the X-ray structure and atomic structure of this GLIC receptor, we were able, Changeux and Delarue, essentially to have the X-ray structure of GLIC closed and GLIC open, and show that we have some kind of quaternary change of the receptor molecule, a twist and some blooming, and therefore have a way to connect the neurotransmitter binding site with the ion channel. And by molecular dynamic studies, Marco Cecchini was able to simulate at the atomic level the conformational transition taking place under this condition, not only for the bacterial receptor, but also for the glycine receptor, and recently for the acetylcholine receptor. So we have here a scheme, if you remember the slide on allosteric, which is highly similar to the model, two-state model I presented by Monod, Wyman, Changeux, which shows the end of my thesis work. So interestingly, we have here some kind of demonstration of the generalization of the model to receptors, even if it is, as you shall see, a little bit more complex than that. So this bacterial origin of our brain receptor and the dynamics of their transition unexpectedly may have some bottom-up effect, which are very significant. Of course, since the network is a chemical network and receptors are also part of the chemical network, they will impose their dynamics to the processing of information in our brain. And when I say that to some colleagues in informatics, they say, oh, what are you dreaming? I say, we have a very slow brain compared to the computers, which are operating at the speed of light. And what makes it slow is simply the allosteric transition of the receptor and the ion channels in the propagation of nerve impulse. So thank you very much. And therefore, we are reaching a rather unexpected conclusion from this work on bacterial receptor and now on brain receptors. And an interesting aspect of these studies is that in addition to the basic allosteric transition between two states, or three states, as you can see here, this is a complexity of the neurotransmitter receptor. There is open, close, and desensitized. Now, in addition to that, there are other types of allosteric interaction involved. And they are due to sites which are localized on the receptor molecule at different levels, and in particular in the transmembrane domain. So sites which are different from the neurotransmitter binding site and the ion channel, and nevertheless modulate the activation process and the opening or closing of the ion channel through activation and desensitization. So these are, of course, pharmacologically very interesting site. And then again, the relationship with applied science is becoming apparent, because one may try to design new drugs acting on these sites. And it was a chance one day with Daniel Bertrand that we decided to try ivermectin, which is an anti-helmintic, which was developed by Omura and Campbell, and for which they actually received the Nobel Prize. But in their Nobel Prize lectures, they did not mention any kind of molecular mechanism for the action of this important drug. And we tested it on the human nicotinic receptor, as you can see here. And amazingly, there is a strong potentiation of the response to acetylcholine by ivermectin. And this response here is, I can show it here or whatever, but it doesn't work, is blocked by mutation in the transmembrane domain. So the conclusion we reach is that some of these allosteric modulatory sites, not all of them, some are in the synaptic domain, other in the cytoplasmic domain, but here we have a very interesting site where ivermectin can bind, which is in the transmembrane domain at a place which is also in relation with the lipids from the bilayer. So we have here a very interesting place to have a modulatory site of a drug acting on not only the nicotinic receptor, but the glutamate receptor, which is involved in the motor behavior of Caenorhabditis, of the worm. So it's a new strategy for drug design to target allosteric modulatory sites. And this is just to illustrate the practical importance of this kind of work, is that there are now about 90 marketed allosteric medicine which are involved with HIV, cancer, hypertension, angina, anxiety, insomnia, bipolar disorders, and so on and so forth. And many of them are acting on brain receptors. And this is, of course, some kind of achievement to have in our hand the atomic basis for the action of these modulators. Now, we'll move to two other parts of my presentation, which is one on synapse selection, the other consciousness. Now, what I said up to now was at the molecular and atomic level, so that we can understand, I believe, signal transduction in the brain, not only at the molecular, but at the atomic level, and resolve through atomic analysis and molecular dynamics the kinetics, dynamic of the transition between the resting, active, and desensitized states. But of course, this is not enough for us, and we want to go a step further in the analysis of networks. And the question which is very important for all of us is the emergence of culture and the diversity of cultures. So there must be some relationship between the environment and the brain. And we proposed many years ago that this could happen as a regulation of synaptogenesis in the postnatal period of brain development, which is especially long in humans, as you know, between 15 and 18 years, and much shorter in monkeys. And you can see that on the left, the brain of a newborn is 5 times smaller than that of the adult. So it means that almost half of the total amount of synapses existing in our brain is developing in this postnatal period. And the rhythm of synaptogenesis is extremely fast, could be at least 1 million of synapses per second. So when you look at your grandchild or child, I'm a grandfather, so that's for the grandchild. well, for 1 second, then 1 million synapses were established. This is something extraordinary, and the white matter is also developing, and there are some candidate genetic events which are involved in the hominization process to extend this long period of development. And this is still a very interesting problem of genetics now, which is to try to understand how this kind of molecular biology at the gene level is taking place. There are some regulatory events. And just as an example, in autism, which is a well-known disease, which corresponds to altered brain maturation, there are about 200 to 300 predisposition genes. So how to solve the problem? I think this is the next generation of scientists after our generation disappear, which I hope we'll be able to respond to this question, which is of course the most pressing question. Now the scheme which we propose with Courage and Danchin is illustrated on the left side of the slide, where you see that there is not a one-to-one relationship between the growing nerve and the target at the beginning, and there is therefore, as a consequence, some kind of overlap between target neurons and many connections being established there. Then the activity of the network is such that there is an elimination of the synapse and a selection which is taking place at this critical stage of development. Now there is a pruning, and we believe that all these steps are interlinked with each other. There is some kind of nesting between these different steps of synaptic development. Now we can have molecular mechanism for this selection of synapses. Some molecular components are identified which are common with all period of similar development in monkey or even in mice. So these are some kind of universal molecular components which are involved in the epigenesis by synapse selection. And what we propose is that since this synapse elimination is associated with the activity of the network coming from the outside or from the inside, then there would be a way to not only store information from the outside, learning, and produce some kind of information collectively at the society level. So this is just to illustrate that indeed some synapse elimination is taking place to support this selection of synapse. Here on the left, you have a development of axons in tadpole, whatever they are, which, as you see, lead to a small number of synapses. If you block the actual development through asynchrony, then there are much more synapses which are being formed. And of course, the activity is essential to eliminate these connections. And on the right part of the slide, you can see that there is indeed, if you look at the total number of synapses, a dramatic decrease of the total number of synapses in the human brain taking place after, let's say, 4 years of age. So there is unambiguously some kind of synapse elimination involved in the development of the human brain. And our proposal is that they are connected with the storage of information from the physical, social, and cultural world around the developing baby, and then has to be relearned at each generation. Now, I just show an example for the selection of cultural circuits, which is here the observation by Desjardins that some lesion in the white matter of the human adult can give alexia. The subject does not read, but is still able to write. So it means that we have a selectivity of the circuit in the white matter, which is under direct control of reading and writing. And as you know, this is not an innate process, since writing has been invented about 8,000 years ago in Mesopotamia. So this is further illustrated by the work of several groups. And here you can see that in the prefrontal cortex of the literate subject, there is much more complexity than in the illiterate. And this is early observation. And a recent one by HUN, you see that this is a very sophisticated change of the whole brain connectivity due to the learning of reading and writing. So there is clearly some kind of relationship between cultural environment and our brain through the selection of circuits in the brain and the whole brain. And just to illustrate the diversity of culture which may emerge from this kind of process, and in particular in reading and writing from Occidental and Oriental countries. So there is a transmission of culture from one generation to the other, not through the genetic equipment, but through relearning at each generation. Now I will end with some more recent work we did on conscious processing, and say that you may not like to use the word consciousness in the mouth of a neurobiologist, but we have tried. This is, again, some definition that there are several hierarchical levels of conscious processing, states of vigilance, use wakefulness, sleep, dreams, global integration of several sensory cognitive memory function and its subjective experience, the capacity to interact socially with self and other, development of culture, and what Spinoza called the drive to survive, which he referred to as conatus, which is some kind of inner consciousness force, which fits very nicely with the presentation which was made before me, and go to the drive to imaginate a new world. So the architecture of consciousness is still not well established. There are some vertical architecture which regulate state of wakefulness and sleep. They are due to thalamocortical loops, vertical one. Now there is also another process which is more puzzling for us, is that there are also horizontal connections in the cerebral cortex which may play a critical role, not in wakefulness or dream, but in the content of consciousness. And the proposal we've made with Stanislas Dehaene is that there are horizontal neuronal connections which contribute to what may be called the conscious workspace. And these long-range axon connection have been already seen by Ramon y Cajal. They have also been seen by Dejrin, already mentioned, and by Patricia Godman-Rakish, where you can see here the connection between prefrontal, temporoparietal, cingulate loci, and this is of importance to link also cognition to emotion. And this kind of network we propose with Stanislas could play the role of a global conscious workspace, and would receive information and give information to unconscious processors like sensory or motor command. Now this is not simply an artifact of my mind, if I may say, but has some reality. And you can see on the right some brain imaging for conscious versus non-conscious processing, and the contribution of prefrontal cortex and parietal temporal areas in what we call ignition, when stimulus becomes conscious. And on the extreme right, there is no ignition, and the signal remains subliminal. So we believe that we have at least some correlates for the access to consciousness. I don't want to say that we have solved the problem of consciousness, but at least we have ways to look at it. And on the left, you have a detail of what ignition could be. And you can see that the time scale is rather surprising. It's 300 milliseconds, which is rather slow to have access to consciousness, but this seems to be the case. And you can also see that in addition to this P300 wave, there are self-persistence of information during this ignition process. Now you have also another example in the monkey, where on the left, this reminds me of Professor Goulash's early work. We have here the visual V1 response to a given sensory stimulus. V4, still higher, still the same. And then when you reach the prefrontal cortex, an ignition which is taking place, and where you see this P300 and self-sustained activity. So this means that this is not simply a theory, but that clearly we can have some, I would say, signs of conscious access in the brain. Now, this is also, of course, happening for more complex situation when you ask a child to recognize a face and appreciate it or not. And there is some signature. This is a work I would like to prefer to all of them. A signature of conscious processing of visual perception. Now, I will illustrate with the next 5 minutes and finish. The further work that we did after identification of the receptor, the epigenesis, and consciousness, to try to understand two things, and I will be very brief on that. Nicotine as a drug of abuse, as you all know. And then in the mice, there is a self-administration of nicotine which can be recorded. This was the work of Marina Picciotto. And if you delete the beta-2 subunit of the nicotinic receptor, as you see on the right, wild type, WT, and beta-2 minus, it means that deletion of the nicotinic receptor and loss of nicotinic receptor in the cortex, the animal survives, but the self-administration is lost. Now this can be further studied, just again as one example among many others, that one can re-express the gene for beta-2 in the mutant and recover the self-administration when it is re-expressed in the ventral tegmental area. So this is an area which is involved in nicotine addiction. So there is clearly a molecular determinant of this kind of operation, and here are some of the subunit composition, alpha 4, alpha 5, alpha 6, beta 2, which are involved in the addiction to nicotine. Therefore, there is a new pharmacology of nicotine which is coming up from these basic science studies, and with the possibility to have ligands which are subunit specific in these receptor oligomers. Now, I will close on one slide about schizophrenia. This is, of course, a very complex and difficult issue. I just want to say that we add a drop of knowledge to this immense problem, which has been studied in many laboratories under different aspects. This is one aspect. First of all, you know from imaging data that in patients there is some deficit of frontal cortex activation. There is a hypofunctioning of the prefrontal cortex. And this is one thing. In addition, there are some genetic predispositions to schizophrenia. Among the 200 to 400 genes which predispose to schizophrenia, we selected these SNPs, which concern the alpha 5 and alpha 3 genes. Now, this led, and I'm being very brief on that because I don't want to bother you too much, but just to say that these work on genome that we did not do ourselves, of course, but which result from a cooperation, as Professor Guyash mentioned, between geneticists and molecular biologists as us and behavioral scientists, that one can show that these nicotinic receptor alpha 5 are present exclusively on inhibitory neurons in the cortex. And the idea is that the inhibitory neurons inhibit the firing of long axon pyramidal cell and therefore alter conscious processing. This is just a rather wild hypothesis, but just to show that future work should continue on these topics and lead us to a better understanding of how our brain functions. Now, in conclusion, we should say that we are facing now a new world of receptor diversity in the human brain, receptor function and so on, acting from bottom-up effects up to cognitive function. And the new pharmacology that one can develop and try to further discover in the future would not only to recognize a given receptor, a given site on the receptor, but a given conformation of the receptor, and therefore have a conformation-specific pharmacology which would let us predict agonists, antagonists, and desensitizer. So we think this is a more refined pharmacology, which is not in our hands, thanks to this basic work. Thank you very much. And before closing, I just wish to illustrate the issue that I have tried to illustrate during this presentation. How one can go from the molecular level to the cellular level, to the network level, to the network of network level, to conscious processing, rational thinking, language, and social life. Thank you very much.
Professzor úr, köszönjük. Thank you, Professor Changeux. Elnök úr, azt hiszem, hogy egy kis meglepetéssel készült.
Jean-Pierre, this is now a very private moment. I have to tell you, this is really private because we have been knowing each other for actually
30 years.
more than 30. More than 30. Yeah. I remember the first conference we met in Brussels with János Szentagotai in 1985.
85, yes.
So it's
35.
almost 40 years.
40 years.
And our friendship goes back almost for 40 years. Allow me to give you a little present as one of the founders of molecular biology, with special regard to molecular biology and the brain. And we had a common friend, one of the real founders of molecular biology, Sidney Brenner. And Sidney was
indeed.
my real, I would say, buddy in Singapore. So I asked the Hungarian artist, László Mátyasi, to make this medal and allow me to give you, on my behalf, but also on behalf of the Hungarian research community and our Hungarian research network, this little medal remembering, of course, to Sidney Brenner, but also your visits to Hungary and your future visits to Hungary. Thank you so much.
Thank you so much. Thank you very much. For me, it is a most distinguished reward that I can receive from you, Balázs. Since we knew Sidney Brenner, he was an immense molecular biologist, and we loved the work, but also the person.
The person, yes.
Was absolutely remarkable and extraordinary. Thank you so much.
Thank you, Jean-Pierre. Thank you.